Greetings, students. Mr. Harrison here from school. Just wanted to walk you through our next slideshow for the next unit, which is on the parts of an experiment, also called a scientific investigation. And the whole idea is if we're going to run an experiment, the experiment should probably be a valid one in order for us to get valid results. So if we want to get valid results, there's a few things we got to include and a few things we have to do. First of all, the definition of a controlled experiment, which is the only kind of valid experiment, is an experiment designed to test a hypothesis in order to get you valid results. An experiment must include some things. So in a controlled experiment, if we want valid results, it has to have a few things included. The first thing is that you got to start with a question or a problem. So a question you want to answer or a problem you want to solve. Any question that needs an answer, a problem that needs a solution would work as long as we can find an answer by the end. And our proposed answer to the problem or to the question is called a hypothesis. That is an educated guess. It is logical, and it addresses what our original question or problem is. And the definition is an educated guess concerning how or why a phenomenon occurs. It should answer your question. And one extra thing, any valid hypothesis must be testable. In other words, it can't just be something that we think. It has to be something that we think that we can also test to find out if we were right. If it's not testable, it's just your opinion. One example that I like to use is if I think, because I am a Minnesota Vikings fan, that purple is the best color, then if I put that as my hypothesis, purple is the best color, that is an educated guess based on my experience. It is logical to me, but the problem is that's not testable. It's just my opinion. Now, if I switch that hypothesis to students in my science class will prefer purple over red, that is testable because I could send you out a survey, gather the results, and either confirm or possibly rewrite my hypothesis based on the data that I get. But it has to be testable. So once we have a question or a problem, we've identified it as a question or a problem, and then we've put forth a hypothesis that addresses that question or problem, we are ready for some change because change can be good sometimes. In a valid experiment, exactly two things, no more, no less. Exactly two things in an experiment are allowed to change. The first is the independent variable, which you see in yellow right here. The second is the dependent variable, which is written in red. And I color-coded these for a reason so that it'd be easier to navigate through this. The independent variable is the variable that is purposely changed, purposely, like on purpose, intentionally, by the experimenter. And that's the variable I change. If I'm the scientist, the independent variable is the variable that I change. This is also the independent variable is also the variable that changes first in the experiment. It's the first thing that we're going to see that's allowed to change. And in terms of a cause and effect relationship, the independent variable would be the cause. It's the first change, so it's the cause, and it's supposed to change something later on in the experiment. The dependent variable, which is in red, is the part of the experiment that changes as a result of the independent variable. It's supposed to be directly because of the independent variable. This is the variable that changes typically towards the end of an experiment, as opposed to towards the beginning, because the independent variable needs the dependent variable needs the independent variable to change first. And in the cause and effect relationship, the dependent variable is the effect of the first change, which is your cause of the independent variable. A few more notes on these two variables. This is a little trick that I've found valuable for me and for some students. Independent variable, if you follow my cursor over here, starts with an I. That's because it's what I change. It's also what I intentionally introduce initially in the experiment. If I were to translate these words into something a little more simple, it would be, it's what I am putting in on purpose at the start, but intentionally introduce initially all starts with I. So think in terms of what am I introducing to the scenario by myself intentionally and at the beginning initially. And I kind of think of it as this is the independent variable, sort of like the whole point of the experiment. Like, what is it that we're testing? That's probably your independent variable. The dependent variable is called the dependent variable because it depends on the independent variable. It depends on this first change over here. It won't change at all unless we introduce this change. And this is usually what's measured and recorded or graphed as the results of the experiment. So the dependent variable is typically included in your results portion of the experiment. And if we were to simplify it a bunch, it would be basically what we found out. The dependent variable is what we found out when we introduced the independent variable first. All right, so there's two variables, independent and dependent variable. There are also two groups. You've got your experimental group for number 1 and your control group for number 2. The experimental group is the group that gets exposed to the change, namely the independent variable. The control group is the group that does not get exposed to the change, namely the independent variable. What I mean by that is if we accept that our independent variable is the change we're introducing, the experimental group gets our independent variable change, and the control group does not get our independent variable change. So the experimental group is the group that gets the special factor, the new treatment, the whatever it was that we're putting into the equation here that wasn't already happening naturally. Where the control group does not get that special factor, does not get the special treatment, does not get whatever it was that we're introducing that wouldn't happen naturally. So it's basically whatever would be happening without our independent variable in the mix at all. That's the control group. The results of the control group are compared to the results of the experimental group to see how things have changed. So the purpose of our control group is for comparison between the group that got the independent variable change and what would normally regularly happen. Speaking of those words, the key words, like the buzzwords for the control group, you're going to be looking for words like the usual group, the normal group, the regular group, the standard group. Those are all kind of synonyms in terms of an experiment: usual, normal, regular, standard. So the way I think of it is that the control group is the group that shows us how things would have been if we hadn't even introduced our independent variable. If we hadn't even conducted our experiment, this is what would have happened naturally without us getting involved. And that is the control group is what happens naturally. And there's one more part that you need to be able to identify in addition to the independent and dependent variable and the experimental group and the control group. And that is this word in green here, constants. So in an experiment, we want to make sure that the only possible thing that could change our dependent variable at the end is only the independent variable at the beginning. We want to make sure that this yellow independent variable is causing the change in the dependent variable and nothing else could be involved. So in order to make sure that nothing else can affect our results except for the factor we're testing, which is the independent variable, and by the way, that's abbreviated IV, or dependent variable is abbreviated DV, in case you got confused. So in order to make sure nothing else can affect our results except for the thing we're testing for, which is the IV, we insert constants, and those are factors in the experiment that are kept the same for each trial. The constants, this is a typo here. Don't worry about this last part. But the constants are factors in the experiment that are kept the same. So those are essentially like when you see the word same, that's a good clue that it's the constant that they're talking about. It'll be things like, let's say we're testing plants to see what's something that might happen to plants. In each of our trials, we're going to give each plant the same amount of sunlight, the same amount of water. They're going to be planted in the same soil, in the same size pot. They're going to be the same type of plant. Like, we want to make sure that the only thing that could change the plant growth usually is going to be the dependent variable in an experiment like that. The only thing that could change the height or the growth of the plant is whatever it is that we're introducing intentionally or on purpose. And so we keep everything else in the experiment constant. All right. So there are a few other parts you need to know, but you don't need to be able to identify them. This will help you on like the vocabulary portion of this module unit. But it is something you need to know, especially if we're going to be able to run our own experiments. So yes, you do need to know the dependent and independent variable as the two things that can change. You do need to know the the control group and the experimental group as the two different groups in the experiment. And you need to know what the constants are for each trial. But you also need to know what a hypothesis is. We talked about that earlier. That's a testable guess that answers our question or solves our problem. It has to be testable. Then you need to know what the conclusion is, and that is a summary of the outcome of the experiment. And basically, it explains whether or not your hypothesis was actually supported by data and why you believe that. And by the way, it's absolutely fine in a science experiment if your hypothesis is not supported by the data. Then you would write up a valid conclusion that says conclusively, my guess wasn't correct. I need to make another guess. And then you make a new hypothesis and test that one. It's perfectly fine. I know that's weird to think of, like you can be wrong in school and that's okay, but it is the case with a conclusion and a hypothesis and an experiment. You can be wrong and have it be totally fine, but that just means you know what the answer isn't, and you still need to find out what the answer is. So then the next part is data. That's just the information you gathered. That's like the numbers or the graph or just the stuff that proves or validates or invalidates your hypothesis. Then number 4, repeated trials. And that's just the number of trials you do. And the idea is the more trials you do, the more accurate your results are and the more valid your hypothesis is or isn't. So if you do the one trial in the experiment, that's good. And the results are typically not invalid, but they're more valid if you do it twice. And they're even more valid if you do it 3 times. And they're even more valid if you repeat it 10 times. So the more times you do it, the more valid your results are. And that's the idea of repeated trials. All right, you did it. Congratulations. Don't forget to answer the Canvas quiz questions that are attached to this post in Canvas. And go ahead and look them up in the slideshow. Don't be too proud to look back at the slideshow and look up answers.